want to take a look at the slide of your ovary. Probably will have a couple of slides of the ovary in your boxes. What I want you to be able to do, for starters, is make sure you can differentiate the various levels of maturation of the ovary. Okay, we're going to start off with, out on the outer edges, you'll see these little circular structures. These are what we would call the primordial follicles. Okay, primordial follicles. We'll talk about the detailed maturation of the oocyte, of the egg essentially, and the different stages of maturation. But just to start off with, looking on the slide, you should be able to identify these primordial follicles, and when they start to mature, if you don't break your slides, they mature into a primary follicle. So here's what we're seeing in a primary follicle right here. Okay, so you see the maturation, the thickening of the outer wall, the growth of the primary oocyte inside of the follicle. As it starts to progress and mature more, you're going to have follicles all over on your slide. You're going to see something like this. You see the follicles getting much bigger. There's the oocyte in the middle, and you start to see a little bit of space forming. It's actually a fluid-filled space called the antrum. Okay, so it starts to form, and this kind of marks how we would identify a secondary follicle. Okay, and so that's a good example of a secondary follicle. And from that point, then the antrum keeps growing, the oocyte matures, and you'll have something like this eventually. This is what we would call a Graafian or fully matured follicle. Inside now we have a secondary oocyte that's ready to be released. Okay, and it's going to be released into the uterine tubes and transported. This is a full antrum. You see the secondary oocyte inside, and this is known as a Graafian follicle. Like I said, the Graafian follicle is ready to be released, so it's going to actually migrate to the edge of the ovary, and the egg inside the secondary oocyte is going to be released into the uterine tubes at that point. After the secondary oocyte is released, then you'll get leftover byproducts of this Graafian follicle that we'll call the corpus albicans. And I forgot about that slide. The corpus luteum to the corpus albicans is also something that you want to take a look at. Because what happens is the Graafian follicle, the follicle releases the egg, and then that follicle turns into eventually scar tissue. But before it turns into scar tissue, it's actually going to be active hormone-producing tissue. Well, let me see if I can find that. Okay, so it covers up the whole screen on here. You're going to look on using scanning or low power. But this whole structure is what we would call a corpus luteum. This whole thing right here. And again, basically it's the leftover remnants of your Graafian follicle, and it becomes hormone-producing or hormone-secreting structure out of the ovary to kind of prepare the uterus for implantation of a fertilized egg.
Did you say it was corpus albicans?
Corpus luteum is what the structure is. Once it becomes deactivated, once it's no longer producing hormones, then it becomes corpus albicans. So initially it's a corpus luteum, and then once it's done producing hormones, it turns into scar tissue called the corpus albicans. Okay, so it goes Graafian follicle, corpus luteum to corpus albicans.
And it produces both hormones?
The corpus luteum does. Progesterones and estrogens. And again, the main point of the corpus luteum is to produce those hormones to get the uterus prepared for implantation of a fertilized egg. Okay, and we'll go through all those cycles in lecture. All right, so those are the two slides you want to look at. For the female, make sure you can identify the different parts of, or the different levels or maturation stages of the follicles. On the male, you got one slide of the testes. You should, and basically all you're going to see is something like this. You should be able to differentiate the seminiferous tubules of the testes from the ducts of the epididymis. Okay, and so again, on scanning power, you'll be looking this, and you should be able to see both of these sections in one field of view. But again, you got the seminiferous tubules of your testes, and then you have the ductules in the epididymis. So you'll be able to see that on one slide.
What are the main anatomical differences as far as the tissues, the
Tissues are going to look very similar. That's when you look at it, in the epididymis, the tubes tend to be more pronounced. Because when you get into the seminiferous tubules, there's a lot of active tissue that's producing sperm. So you have thicker edges, more nourishing tissue. So you see thicker walls typically.
Are those cuboidal?
There's really no distinguishment because it's not epithelial tissue per se. But yeah, if you look at them, you'll see some cuboidal, you'll see some squamous. Again, and there's connective tissue in there as well. So there's really no identification as far as the specific shape of the cells. Because these are not epithelial linings. These are all inside. And then the epididymal ducts, they lead to the ductus deferens. Okay, so we got that one, and then the last one, which you'll have to look at under high power, right there. So here's sperm. There's your sperm. When we go all the way up to high power, it's kind of hard to focus on just one because there's a whole bunch of them. But when you go all the way up to high power, you can kind of focus on one. You see the flagella, you see the acrosome, you see the midpiece right here. So really just identify the head, the midpiece, and then the flagella. Okay, you can kind of make that out. The head with the acrosome, the midpiece, and then there's the flagella there. Okay, and so that's really it, it's sperm, and then the three main parts of the sperm. We do have the models, so make sure you look at the models and you can identify all the detailed structures. So there's 4 slides today, and then plus the digestive slide. So we got 5 slides total you want to look at. Again, I mentioned this before, we got a lot of models, so most of your lab is going to be made up of the models of digestion and reproduction, and some diagrams as well. Okay, with a couple of slides and maybe a couple of cats. Okay, so make sure you're focusing your attention on those models. All right, so that's what you're doing today.